Talking about the based on function, based on the function. So based on the function, basically we are classifying the main thing behind classifying all this into the 6 type is that bacteria prefer different not a function, different types of nutrient, different types of nutrients. That's why we have the 6 classification. Now talking about the first, enriched media. Second one is the enrichment media. Never ever confuse, both are entirely different. Although the spelling are same, but the use and the chemical constitution, both of them is different. First one is the enriched media. Example is the blood agar and chocolate agar. What we are doing is we are taking the what is called as the petri dish. Okay, we are putting the samples and we are giving them the sheep blood or horse blood. Okay, because the bacteria which is growing here, you can write down the examples. It's a streptococci. Streptococci, and it is for the blood agar, and for the chocolate agar you remember is Neisseria gonorrhea and gonorrhea. Okay, Neisseria gonorrhea, which is basically causing the gonorrhea, the sexual transmitted disease. Okay, now the thing is this streptococci, it need the specific nutrient. So like हम लोग तो मजबूर हैं, हम लोग तो देना पड़ेगा, जो मांगेगा वो देना पड़ेगा, नहीं तो हम लोग उसे देख ही नहीं पाएंगे. तो देखने के लिए जो मांग रहा है वो देना है. तो क्या ये मांगता है? It's nothing more demanding. The simple things, we are providing the blood. First of all, what blood? It's a sheep blood. Okay, sheep blood देते हैं, and apart from that we are giving here basically the protein source. What source? Protein source we are giving here. So for the protein source, it can be the eggs. Okay, we can give the eggs yolk, which is basically protein source. Fine. Now then we prepare the blood agar. By giving all these things, we prepare the blood agar, and we are able to see the streptococci. Okay, streptococci. Talking about the chocolate agar, although it is not chocolate in color, remember the color also. The enrichment media, it's of course the blood agar, so it is like a blood color. Okay, blood color. If we're talking about the chocolate agar, it's a brown in color. Chocolate agar is a brown in color. Why it is brown in color? Because the same things we are giving, the same sheep blood we are giving into the chocolate media also, but this time this blood is heated. This is the heated blood. The sheep blood first we will heat, and then we will put into that what is called as the petri dish, and we will make the chocolate agar. The thing is, when we are giving this gonorrhea bacteria here, they will start hemolyting the blood. Hemolyting means, you know that any organism blood there is a presence of hemoglobin. There is a presence of hemoglobin. Fine. When this bacteria start growing, they will break down the hemoglobin. That is called as the hemolysis. Lysis means breakdown. Hemo means blood. It is the breakdown of blood. So when blood will break down, it will change the color into the chocolate color. Okay, brownish color. Got the point? Yes? Everyone? Yes. Got the point? So that is why it is called as the chocolate in color. This is not a chocolate actual. Just because of the color, we are giving the name. Clear? Fine. Remember the example 2. Now coming to the enrichment media. See, the similarities is there between the enrichment media and the selective media. Enrichment media is liquid media. It is a liquid media, whereas selective media is a solid media. Are you getting the point? Okay. Enrichment media is the solid media and selective media is the liquid media. Okay, now it means you are listening. I am not concentrating. Fine. So enrichment media is the liquid media and selective media is a how to remember? Simple, as for the selective and as for the solid. Easy to remember? Fine. Yeah. What we are doing here? Nothing more. When we are taking the sample, any bacterial sample from blood, serum, sputum, fecal matter or urine, that is basically a mixture. We already have the concept, and that is, in our body there are microorganisms which is normally present, which is not basically harming us, and they are helping us. That is called as the normal flora or microflora. These are the group of microorganisms which is present inside our body, and they are helping us to perform day-to-day activities, the inside activities which is result into the outside activities. The one thing is that कि इंसान जो अंदर से रहता है वो बाहर से भी दिख जाता है, isn't it? जो इंसान अंदर से है नेचर, वो बाहर से भी वो एग्जिबिट होता है. इफ यू आर इंटरनली फीलिंग वेल, काम, देन आपका एक्सटर्नल अपीयरेंस भी वैसा ही रहता है. आर यू गेटिंग माय पॉइंट्स? So the bacteria which is present inside us, they are basically internally helping us in so many ways. Already we discussed. Fine. And that is exhibiting outward also. We are feeling ourselves good and fine and able to perform the day-to-day activity. Hence, we can state that this microflora helping us to drive or channelize our day-to-day activities. The thing is, when we take the samples, along with the pathogenic bacteria, this normal flora also comes out. The normal flora also comes out. And when we are putting on the nutrient media, along with the pathogenic microorganisms, this normal flora also will grow. Okay? They will also grow. But हमारा काम क्या है? हमको तो pathogenic bacteria चाहिए. अगर बहुत सारा bacteria grow हो जाएगा, then it is very difficult to identify or label which is the pathogenic and which is the non-pathogenic. तो हमको क्या करना पड़ेगा? यहाँ पे दिमाग लगाना पड़ेगा. दिमाग लगाने में हम क्या दिमाग लगा सकते हैं? तुम लोग बताओ. इसमें दो तरह का बैक्टीरिया आ गया, एक पैथोजेनिक है, एक नॉन पैथोजेनिक है. हमें चाहिए पैथोजेनिक बैक्टीरिया. हम क्या करेंगे? तुम्हारा दो फ्रेंड है, एक गाड़ी बुक हुआ, ठीक है? यू हैव टू फ्रेंड, यू बुक द वन कैब, बट यू वांट your one friend to come, another friend not to come. What you will do? Common question, no? एक को रोक लेंगे, या रोकने के लिए तो कुछ बहाना बताना पड़ेगा. The same things what we are doing here also. The thing is कि इसके अंदर में हम लोग कुछ इनहिबिटरी चीज डालेंगे जो नॉर्मल फ्लोरा का ग्रोथ को इनहिबिट कर देगा. Called as the inhibitory substance, inhibitory chemical substance. What they will do? They will basically suppress the normal flora. Okay? और हम लोग न्यूट्रिएंट के थ्रू जो बैक्टीरिया हमें चाहिए उसको ग्रो करेंगे. Clear? This is called as the enrichment media. Example you remember. It's the tetrathionate broth. Again broth. It means it is in a liquid phase. It is in a liquid state. No problem. Coming to the selective media. The same things here what we are doing. Same thing. Basically suppressing the undesired microbes and enhancing the desired microbes. So here we are more, you can say, चालाक. Here हम लोग अपना ज़्यादा दिमाग लगाते हैं. हम लोग जल्दी के लिए क्या करते हैं? तो पहला तो इनहिबिटरी सब्सटेंस डालते हैं कि जो नॉर्मल बैक्टीरिया है वो ग्रो नहीं करे. जो पैथोजेनिक बैक्टीरिया उसके लिए हम लोग स्टिमुलेटरी सब्सटेंस डालते हैं. What we are providing is the stimulatory thing जो उसके ग्रोथ को और सपोर्ट करे, उसे ग्रोथ को और सपोर्ट करे. ये दो चीज एग्जाम तुम याद रखो. एंड अनदर थिंग व्हिच यू हैव टू रिमेंबर इज दैट The enrichment media is in the solid, no, it's in a liquid state, and selective media is in a solid state. Fine. And the example of the selective media is the McConkey agar. Example is the McConkey agar. No problem. Yes. Yes. और एक बार समझाएं. Fine. No problem. So we are discussing the enrichment media and the selective media. Fine. We said that enrichment media is in a liquid state and selective media in a solid state. But both serve for the same purpose. Both serve for the same goal. The ultimate goal is what? To promote the pathogenic bacteria, what desired bacteria we can say. What we deciding, किसको grow करना है, क्या हमारा इच्छा है, okay, promote the desired bacteria and inhibit the undesired bacteria. So for that, we are in the culture media, along with all the whatever nutrient they needed, we are giving the inhibitory substance to inhibit or suppress the undesired bacterial growth and promote the desired bacterial growth. Example is the tetrathionate broth and the McConkey agar. This is enrichment media is commonly used. It is commonly used for the identifying the diarrheogenic bacteria. Diarrheogenic bacteria means all those E. coli, okay? E. coli bacteria. Talking about the selective media, we are providing the McConkey agar. McConkey agar. And this selective media is when we have to differentiate between the two microorganisms. I mean, there is a mixture of things is present in the sample, the blood which we are going to take from the patient, mixture of the bacteria is present. But when we have to clear cut the bacterial, you can say the colonies, on that time we are providing the selective, using the selective media. Now coming to the indicator media. Indicator media, here what is going to we do is that we are putting some chemicals in this media. We are putting the chemicals in this media because the colonies which are forming, is not as visual as you can say, जिसे हम लोग अपने आंखों से देख सके. इतना विजुअल नहीं है कि हम लोग अपने विजन में उसको अच्छे से देख पाए. ठीक है. कुछ कुछ बैक्टीरिया स्लो ग्रो करता है, कुछ कुछ फास्ट ग्रो करता है. तो वो ग्रो करे और उसे हम देख पाए, ठीक है, उसके लिए हम लोग क्या करते हैं, तो इंडिकेटर मीडिया को यूज करते हैं. वी आर एडिंग द केमिकल हेयर, और जैसे ही बैक्टीरिया ग्रो करेगा तो इसका जो कलर होता है, सी ऑल द मीडिया शोज द वन काइंड ऑफ कलर. ओके, कलर किया हमने, इसमें न्यूट्रिएंट डाल दिया है, बैक्टीरिया थोड़ा थोड़ा ग्रो कर रहा है, तो थोड़ा थोड़ा हमें दिख रहा है. बट व्हेन वी आर एडिंग द केमिकल्स, बेसिकली हम लोग यहाँ पे एक केमिकल डालते हैं जो क्या करता है, तो जैसे ही बैक्टीरिया ग्रो करेगा तो इसका कलर चेंज कर देगा. ठीक है, द कलर ऑफ मीडिया विल बी चेंज. देन हमको पता चलेगा कि बैक्टीरियल ग्रोथ इज गोइंग ऑन, बैक्टीरियल ग्रोथ इज गोइंग ऑन. Got the point? No problem. And the example you remember is the mannitol salt agar. Agar? Yeah, mannitol salt agar. No problem. Differential media, we already discussed that. Thiocyanate. एक ट्यूब लेते हैं, ट्यूब के अंदर में वी आर गिविंग थायोग्लाइकोलेट, वी आर पुटिंग, ओके, वी आर पुटिंग द agar, we are also you can say putting the nutrient source, isn't it? We discussed, no, and then we are able to differentiate the bacteria. So your example is the, here the another example I put it is the XLD xylose lysine deoxycholate. Okay, then we have the transport media. What is the use of transport media is that when we are taking the sample, no, sometimes we have to preserve it. Okay, we are not immediately start testing. We have the not of one sample in the lab, we have the so many samples, so it takes time. Okay, तो उस दौरान जितना टाइम लगता है उस दौरान कोई बैक्टीरिया मर नहीं जाए, ठीक है, वो जिंदा रहे. So for that we are using the transport media, and the example is the estuates media. Clear? So this is all about your culture media, and this is very very useful in the laboratory diagnosis, lab diagnosis. Clear? You have any questions? No? Yes? Clear? Yes? Shall we move ahead? Yes? We'll move ahead. The thing is, you have to revise it in order to understand all the things. You have to again and again revise till the examination at least. Fine? Fine. Now let's move ahead and discuss about the standing technique. Standing technique.